I don't know the legalities, and there might be issues around this that I'm not aware of, but they're working on this model of humanitarian licenses as a way to sort of get industry involved. And he mentioned 3 companies that they're working with now: Immunocore, which has an HIV intervention they're developing in the UK, BioNTech, which is a familiar name now because they're one of the manufacturers of one of the COVID mRNA vaccines, and also Novartis, which is a huge company that they're now collaborating with on, I think, both the sickle cell disease and HIV gene therapy initiatives. So it's kind of optimistic that there's sort of industry know-how being brought to this effort as well.
Super to
Michael
hear.
and Richard, we have a follow-up question about the CAR T cells. So the question is, would these CAR T cells be used to mop up the remaining cells after ART reduces the viral load, or would they be a replacement for ART?
So I think the idea is that the goal is to have them be able to be a replacement. But I think, they're ways off from being confident that that's achievable, at this juncture where we are now, but that is the goal. The idea being that these cells would be able to control the virus the way, some rare people like elite controllers are able to do. And so the hope is that somebody would be able to stop antiretroviral therapy and have their immune system and these modified T cells kick in and control any replication.
Right. I think the goal is trying to get some sort of therapy that is without the use of daily pills. Ultimately, there might be, especially in the early days where we might see some success with these interventions, there might be times where we have overlap, but I think they were even hoping that this would be a treatment where we would find people who acquire HIV and they could be given this treatment instead of ART and have one treatment once and a person's taken care of, whether it's totally eliminated from the body or able to control it like an elite controller where it's basically showing no harm. Of course, that's a very pie in the sky dream. They're also dreaming though about what they call in vivo delivery, which is having it all in a syringe that you can inject and you can bring to people wherever they are. Which is still, that's again, very aspirational in terms of a goal.
Well, I see Katie Barr has her hand raised, and Katie would be the expert on this.
Come on in, Katie. Welcome.
Hey, everyone. Such a nice discussion to be a part of. Thank you for inviting me. I just want to say, I think that there's many layers of the aspirations for what CAR T cells could ultimately do. But at a first step, I think the goal is, whereas antiretrovirals protect cells from the next round of infection, they don't actually clear any of the persistently infected cells. And the goal of a CAR T cell is to have this enhanced sort of CD8 T cell that can go and identify infected latent cells, whether that's while you're still on antiretrovirals or after antiretrovirals, and get rid of that reservoir of cells that is currently not impacted at all by our current antiretrovirals. So it's to do that thing that the antiretrovirals can't do at this point. And then, as Michael and Richard said, ultimately that could get to a place where you don't need antiretrovirals anymore because you've reduced the reservoir or eradicated the reservoir enough that you don't need to continue to protect your cells from the next virus that gets sort of spit out. And I'll just say as part, I'm from Penn and we have one of these trials. And, this year has been crazy and difficult for so many reasons, but many of these HIV cure studies, these clinical trials have been paused for months and months and months, of course, because we need to prioritize safety of our participants. And from a clinical trials perspective, they wanted to prioritize, doing COVID studies, doing vaccine trials, et cetera. So there's capacity issues, but some of these are back online. So, we want to maintain sort of a priority for HIV research, even while we also protect our participants and protect our staff workers and, focus on doing as much as we can to eradicate COVID. We're trying to still sort of find some balance where we can get back to doing our clinical research on HIV, because that is still very important to us, even though we have this massive pandemic that's going on right now that we're all trying to also deal with.
Thanks
Katie,
for that. Go ahead, Michael.
I was just going to ask Katie, but while we were talking about Penn specifically, I think it was some latest research. Did you not have a paper recently that pointed out that most of your participants in the study were Black? Was that from Penn, a recent study with Pablo Tebas?
And tell us why that may be perceived as important if that were your study.
Well, do you mean with some of the gene therapy studies?
Yeah, specifically.
So our Philly trials network, actually almost all of our clinical trials, we have a pretty good sort of diverse representation of Philadelphia, which is about 50% African-American, maybe 10 or 15% Hispanic, and we get a pretty good job of representing our community. We have an amazing community group. I know a lot of places do, but ours is really excellent. And so almost all of our clinical trials that I've been involved in are somewhere between 40 and 70% African-American participants. And that's been the case for our recent HIV studies, like our gene therapy studies, our antibody-based cure studies. A lot of our COVID studies, in fact, have had that similar sort of about 60% African-American representation. Now, that doesn't mean we're fully representing everybody in all the ways, but that particular component of our trials, and that is, as you mentioned, Danielle, it's hugely important because the vast majority of our HIV research, particularly in cure, has dramatically underrepresented key portions of the pandemic, whether it's outside of the United States, but within the US, we really struggle in particular with African-American enrollment, but also women. And I sit on the sort of decision-making groups for a lot of these trials, and we sort of try really hard to think about how to increase our enrollment of women, in particular younger women of childbearing potential, or any individuals of childbearing potential, because everybody is so anxious about enrolling someone in a trial who then becomes pregnant and what the consequences are for that person and for the fetus, etc. And so we use these kid gloves that make it so restrictive for any of those people of childbearing potential to actually enroll into one of these studies that it becomes ridiculous. I mean, I think about it all the time. Would I enroll in this study? Heck no, you're asking me to do ridiculous things, just unreasonable things in order to participate. And so I think that's a major issue in HIV clinical trials, is we recognize we need to increase representation of women for 1,000 reasons. And we also want to be protective of women of childbearing potential and their potential children, but we got to find some place in the middle where we make it reasonable for those people to join these trials and not like a third job that they have to do while they're trying to live their lives. Anyway.
I couldn't agree more. And then with that said, we'll take a quick pause. I see Dawn has her hand up so nicely. Dawn?
I'm trying to behave. Yeah, I think that, Katie, I really appreciate you bringing up this issue. This is a perpetual challenge. We've been fighting this for decades now. And this whole kind of, dilemma about whether or not we are protecting women and their children, or whether we are actually hurting women and their children by not doing studies that include them, so that we then force all of the data to occur on the streets and out in the real world, and not in a place where we can collect it. So I think we have to continue to go back to that conversation and look for ways to involve women so that we have information that's real world. And that doesn't mean that we need to be irresponsible in how we do it, but we have to be willing to kind of allow women. And I know this is not, I know you're talking about specifically about cure research, but I think across the board, we need to have some really hard conversations about this.
Look where we are with COVID vaccines right now. It's incredibly challenging. If you're a young woman who's pregnant or considering getting pregnant, or if you're an individual who's pregnant, what are you supposed to do about the vaccine? There is no data. The recommendation is to talk to your doctor. What does your doctor know? Nobody knows.
That is a classic punt. That is like the hot potato. That's what we've done for women for the last 3 decades, is we've said, we can't put you in these clinical trials and figure out how these drugs work. So just go out there and do it on your own with your doctor who has absolutely no idea what's going on. So that, I won't derail our conversation on cure to go into this, because as many of you know, I can go on about this indefinitely, but Judy Currier, Risa Hoffman, Rosie and myself, we just put out an editorial on this in JIAS this month. So
Oh,
I'll
wonderful.
put in the chat for those who are
Congratulations
inclined.
on that, Dawn.
And Dawn, would you mind sending me that link and I'll add it to the follow-up email that we'll send to everybody? And there's also a number of questions that are coming up in the chat. So my moderators, I don't want to you may have a certain sequence of where you want to go with this. Are you ready to take some questions, or do you have more things you want to present before we get to some follow-up questions?
So we're ready for the questions. We can thread them into the discussion. I also have the chat up, and I'm going to impose the questions as they become relevant in the discussion.
And maybe I'll just add one thing before we just end that part. There are studies at this CROI about sex differences related to the HIV reservoir that may be relevant to cure. I haven't been through everything, but I saw at least 2 presentations. One from Eileen Scully's group, who's been doing a lot of work on the issue, and then another looking at the effects of reproductive aging and menopause. So those abstracts are worth looking out for.
And Danielle, just to be clear, you see the questions from Alex and Udam?
Yeah.
Okay, great.
Address Udam's question. Udam, Katie, you were talking about how to safely bring back online some of these cure trials that have been interrupted during COVID. So Udam had a question that talked about during COVID, especially in the surges, when you consider all of the, vacillation between the stay at home, okay, go outside, have dinner at home, go have dinner, at your local place outside. Will ATIs or can ATIs be done safely considering the intense, frequent clinical visits? What are your thoughts?
Yeah, that's a great question. And it's a huge topic of all these trials that are currently figuring out how to navigate in the world of COVID. And as you said, Danielle, the world of COVID shifts every couple of weeks, and it's very different from one site, even within the United States, but certainly internationally, we're looking at really different realities moving forward. In the United States, if you're in a place where we're getting vaccinated more and more over the next few months, that's a very different situation to open up a trial where you think there's a treatment interruption coming in 9 months, as opposed to a lot of our international sites where the prospect for when these vaccinations are going to happen is totally different. So I think there needs to be local assessments about how safe it is to do a treatment interruption for the participants. Can they go back and forth can they get transportation? Can they go back and forth to a study site? For the study site personnel, are they able to work and do all of the HIV ATI stuff when they may be overwhelmed with other sort of capacity issues? For places that are have more access to vaccines, I think we have to think about questions of when do you give a vaccine during a treatment interruption?
Oh, yes.
What about if someone gets a diagnosis, but they're asymptomatic or they have symptoms? How do we deal with those? And my understanding is almost every trial that's moving forward is coming up with a comprehensive plan of how they're going to navigate this setting with the situation right now, moving forward, and what that means for that study. And some sites are saying it's just not reasonable for us to put our participants through this right now. And others are saying we project by the time the treatment interruption for this study would happen in 6 to 24 months, we're going to be in a place where we can do this, and here's the way we're going to structure this. So there's a lot of thoughtfulness in this area, and that's a major issue for any study moving forward right now.
Indeed. Richard, do you want to weigh in here? I have one more follow-up question, and I'll get to some that are also in the chat as well.
Yeah, I would just recommend the papers, with the links. There'll be a way back in the chat now, but I can repost, and I think Jim's going to include them in the email as well, that make some recommendations for how it might be possible to conduct ATIs within the context of the pandemic. Awesome.
Simon had a really good question. It was very provocative in our discussions before, as well as that, is it a reasonable requirement of folks to have been fully vaccinated for COVID before allowing them into ATI trials? What are your thoughts, people?
I mean, we haven't required it because there are some reasons why, people's autonomy should, they should be able to make that decision for themselves. And but we recommend, as we all do, that if you have the opportunity to get that vaccine and it's available to you, that we would certainly delay a study for a period of, a month to 2 months in order to allow you to go through that full vaccination course and get to that place. But if an individual has decided they don't want it or they can't take it for various reasons, then we would try to mitigate risk as much as possible and allow them to participate.
I thought another provocative topic that came up was talking about it offering a COVID vaccine as a part of the study. In other words, if someone was not vaccinated, vaccinating them. And then there was also the comments about that came up about it being coercive or potentially coercive.
Let's be clear.
Yeah, that I don't think any trials group has access to a vaccine to be coercive, but I do think that could potentially be very coercive, and we would not want someone to sign up for a trial that they didn't fully want to do for all X, Y, and Z reasons in order to get this potentially life-saving thing that has been so difficult to access. So yeah.
You're speaking my language, Katie. You're speaking my language. Right, we want to consider individuals' autonomy to participate in clinical trials, and especially given some of the rigorous requirements that are part of ATI trials, offering a vaccine could be the tipping point, right? It's literally a matter of surviving or not surviving in the era of COVID. Sometimes that's what it comes down when you have access to a vaccine. So I think we should consider the center conversations of equity, but I'll get off my soapbox and welcome Julie. Julie, your question? She raised her hand so nicely.
I just was going to play devil's advocate with you, Danielle, because I think it'd be really interesting in our minds to compare it to access to PrEP in prevention trials. I know we've been debating that, and the idea of a trial providing PrEP to people versus saying, can we help you get on PrEP from the community, I think has been this ongoing dialogue, and I think it's very similar. I'd just love to hear people's thoughts about that.
So same context for prevention trials. What are folks' thoughts? And I see your hand, Simon. I'll get to you next. Julie, if you could lower your hand.
And Danielle, this is a follow-up from Jessica. Could a COVID vaccine actually be an exclusion criteria?
And just to add context, the reason why I ask is because it obviously does have an immune reaction. And so when you're talking about some of these immunotherapy responses, there could be interactions that we don't know about that could confound a trial. So
Jessica, I will just comment because I'm much better equipped to answer your questions than the other one. What we've tried to do is think about COVID vaccines like all other vaccines, and we absolutely want participants to get their vaccines whenever it is recommended, whatever, if there's an annual flu shot, if there's a shingles vaccine, whatever. We want you to get that vaccine whenever it is recommended so that we protect your health in the best way possible. But what we could do is slightly shift the study schedule around that. So we're not, so we get you a vaccine on day one, and then we shift that window where we're going to check a viral load back a week. So we're not looking right at the period of time where we expect that reaction. But, standard of care right now is that people get vaccines throughout their year, throughout their life. And we have to figure out how our cure strategies and immunotherapy are going to work within that context. And we have to build that into the construction of how we interpret the results. But it's a good point, Jessica, that we need to factor that in.
Absolutely, great point. We'll stop and take Simon's comment or question.
Hey, thanks. So just for information, the ATI study, the Rio study, which was stopped just about to enroll at the beginning of March last year and has been on hold since, tried really hard to get vaccines as part of the research, as part of the study, and we were in a good position to be able to do that because some of the Rio group also worked for the Oxford group that had produced the vaccine, and we sort of got approval. The researchers and the company would have made a small amount available, but the complexity of adding something without it becoming part of a study was too difficult to get it to be used. Like if you included the COVID vaccination as part of the ATI study, then it becomes something that has to be evaluated as if it's an intervention, which would be terrible. And if you tried to bolt on the participants in the Rio study to the HIV Oxford sub-study, then that was involved too many clinic visits. So people tried really hard in order to do this, in order to get the study to enroll a bit earlier. And then the mention in terms of PrEP for prevention studies, it thinks it's a really hot topic, because the AMP antibody study really is reporting now as if PrEP was available throughout the study to all participants, but it wasn't really. And I think they just trickled through access, particularly in the African study. I think there's been no data about uptake of PrEP in the Southern African countries, but I'd expect it to be really low. And I understand why they sort of staggered it and said we're going to only provide PrEP if it's part of standard of care, but it meant that most people didn't really have access.
I was going to say, could there potentially be more data coming in the future about access or uptake?
It will be coming, yeah, but it was one of the bits of information that was missing from the presentations a week or two ago at the prevention conference.
I was going to be messy, but I'm not going to be messy.
Why not? Come on, bring it.
I know, but I want to be sure that Julie's question is answered. Katie or Richard, would you all like to chime in?
Which question was this? I'm sorry.
Julie, could you please repeat your question?
That was the one really that Simon was addressing, just about this comparison between access to COVID vaccine in a cure trial versus the idea of access to PrEP in a prevention trial. And granted, they're different in that PrEP is, it becomes the standard of prevention. But it's really, that question of being life-saving, I think, is really important, particularly, I think, in light of what Danielle was saying about communities who don't normally have access because of the issue of being coercive. But on the other hand, what does it mean to not offer the standard, particularly if we say, just as Simon was saying, that if it's a standard in a given country, is that or is that not true in the setting of a trial? So thanks.
I will say that there are some nuanced differences because PrEP is both life-saving, but it also directly impacts the study of HIV prevention trials. And so there's this inherent tension. If you are doing this trial in a community where PrEP is maybe theoretically or difficult to access, but theoretically available, but not really used very often, and you think your prevention strategy could actually really make an impact there. But if you enroll people in that study and you give them this, and you somehow increase the rate of usage of prevention, I think that's the tension, although I think we should all be attempting to provide whatever standard of care prevention is currently available to all of our prevention trialists, participants. In the COVID world, we desperately want people to get the vaccine, and it has nothing to do with our cure. In fact, getting the vaccine helps you complete the cure trial and for us to learn about these other things. So there's no inherent conflict there. We just are dealing with issues of access and of this scarce resource that we desperately want people to have access to, but we don't want to force them to take to join, as Danielle said, a really challenging set of, restrictions for these cure trials.
Thank you for that. Richard, can you weigh in there?
No, I would just echo that. Completely agree.
Awesome. So Simon's on fire in the chat saying that COVID vaccines are so widely recommended for the general population that perhaps different to PrEP, which involves continued treatments.
And I did, if it's okay, there was a question, I think, from Alex Schneider earlier about assays. And I don't know if maybe Katie, just asking about.
Was asking about, is there any news about more sensitive testing so we can actually get a more accurate measurement of the reservoir?
QVOA versus Tilda.
Yeah, so I think that one of the things that's happened in the past couple of years in CURE is that the intact proviral DNA assay, which is a high throughput sort of digital droplet PCR assay, which looks at cells from people on antiretrovirals and is able to differentiate between cells infected with defective virus and cells infected with theoretically intact virus. And it's a cheaper assay, it's a faster assay, it's been shown to be pretty reproducible and to correlate with our virus outgrowth assays. It's a much higher estimate. It's clearly still an overestimate of sort of the reservoir, but it's closer to what we think the real answer is. So that assay, it still has issues. It doesn't work as well for every person, and it doesn't work as well in non-clade B virus, which is a huge issue for the whole world. But that assay has been applied now to multiple different sort of large cohorts, and it's shown interesting things. First of all, that it's reproducible across these cohorts for the most part. It's shown that our intact virus actually declines over time a little bit faster than our total reservoir, which includes, for the most part, defective virus. So that gives us a little bit of hope that like our natural immune responses are actually seeing that HIV reservoir and trying to clear it a little bit more than we thought it had. But I do think that assay is, for right now, probably going to become more of a standard for what we use to measure the reservoir across clinical trials, across large cohorts. And that assay needs to be improved upon, so it's more efficient and it's cheaper and it's more accessible to lots of labs around the world. But it's a big step over total HIV DNA, which is what most trials had used previously, which is sort of a huge overestimate of the reservoir that doesn't correlate as well with some of our more sort of gold standard assays like the virus outgrowth assay, or much more lab-based assays of differentiating between intact and defective virus. So I do think the IPDA, this intact proviral DNA assay, is probably going to become what we use for these studies. And hopefully that will allow us to compare between strategies better. So we can say, how does this compare to that with a more even, apples to apples comparison. It still has work to go. It's still not fully accessible to the whole world, but I think it's a step in the right direction. And there are other assays like that are continuing to be developed.
And speaking of other assays, Katie, isn't it in Penn where you are working on a home viral load testing?
Yes,
we are doing studies on home viral load testing, and I just think that is going to be revolutionary if we can get an accurate sort of home test for people, especially if you're undergoing a treatment interruption. Exactly. And you don't want to go back to the clinic every week, every 2 weeks, every 4 weeks. I think that's going to be huge. So we're working on trying to pilot these tests and figure out what works. I think if we actually get some good immunotherapy that leads to sort of a elite controller-like status where you're suppressed for a long period of time, having that assay where people have the capacity at home to test maybe once a week, maybe twice a week, and get a sense of, is my virus really suppressed? Am I safe to be in this you versus you category? Am I comfortable with what this therapy is doing, or do I want to go back on antiretrovirals? It just gives a lot more sort of fine-tuned information as opposed to people having to come into the clinic and get the test, and then the test takes a while to come back, and then you go back to the clinic, etc. So I think it would be huge for clinical trials. It will also be great if we actually get anything that works to allow people a little bit more security to know that this is working for a long period of time. So I'm really hopeful. And I think the technology is there. We just have to work out the kinks and get it sort of tested and then figure out how to best implement it.
Yeah, I agree with what Simon just put in the chat, that it was an amazing overlap with the needs with cure research and global access to viral loads, everything. I think that's a thing that we didn't know that we were really missing until we really started to recognize what we needed. Absolutely. And just to hear you guys working on it, it really gives me heart.
Can you talk about some of the cost considerations of these reservoir testing?
The reservoir testing, like the IPDA, the cost? Yeah, I think it's still fairly expensive. There's a company that does it now, but there are a lot of homegrown versions of this assay because there's a specific platform that you can use. If you have a digital droplet PCR, you can build this thing for yourself. You just need to sort of. So I don't know exactly what the cost is going to be. It's more expensive than an HIV DNA, a total DNA, but I hope that we'll figure out a way to make it really reasonable for groups all over the world to use it in all their studies. In terms of the whole viral load testing, I have no idea. I'm assuming, I mean, it's only really usable if it's inexpensive. And a lot of these sort of devices really make things accessible, but that's it has to be accessible in all ways, cost and accuracy and convenience, etc.
Indeed. So Richard took care to post some of the references to the sex-based considerations in some of the trials. So just so folks know, check the chat box, it's on fire, and it'll also come out after today's discussion. Alain posted in the chat and says, Katie and Richard, do you really see a role for immune checkpoint blockers in the improvement of CD8 and NK cells capacity to kill infected cells? I believe these studies are running.
Richard, do you want to take this one or you want me to?
Maybe, do you want to go first?
Sure.
Sorry,
So,
thank you.
I think there's two sides to this coin. First of all, checkpoint inhibitors in the cancer world have revolutionized the way we treat cancer. And they have led to many people being cured or having years and years and years of high quality life after a diagnosis that previously would have led to, a really early death. So the potential to augment immune responses is there. But as they currently exist, they have a really challenging side effect profile in that most people do okay, but some of the side effects are permanent and they're really potentially debilitating, like some of these autoimmune diseases that can be raised, et cetera. So it's really hard to figure out in their current formulations how we use them in people living with HIV without cancer. Because right now there are studies in people living with HIV and cancer that have shown impressive results, and there are maybe smaller studies. So my hope is that we are going to get new formulations of these checkpoint inhibitors that are slightly have a better side effect profile, and that actually I think could be a real boon to HIV immunotherapy. But as they currently exist, I am skeptical that they will be accepted for people living with HIV who are basically pretty darn healthy and don't want to take the risk of a very serious side effect.
Over to you, Richard.
Yeah, there's one abstract I spotted at the conference where it's an NIH study just giving a single dose. But I would echo the same concerns that in sort of studies with repeat dosing, the toxicity profile just looks like it's going to be impractical. And so I think Sharon Lewin in particular is really keen on trying to look at sort of improved formulations that may have less issues. And I think right now, like what's out there probably isn't something that could be used in an ongoing way as a cure intervention because of the side effects.
There's a ton of information on these in the oncology world, and we can learn a lot from them. And hopefully as the oncology world continues to develop these, we'll be able to, again, steal from them a little bit and get some of these new formulations because they are really important immune modulators. It's just a higher risk situation.
So I'm going to take this pregnant pause, as it were. There was a great question that came in earlier from Jessica, really asking all the presenters today, what are you looking at? What are you going to be checking out at the conference related to cure research? Where would you point people to? What's on your itinerary that you're especially interested in?
Very good question. We can.
I will try to be really fast. I'm going to point out 4 seminars or presentations that I think are really exciting. The first is Dr. Xu Yu is giving a plenary on their group of studies that look at elite controllers and what the reservoir and elite controllers look like. And they have combined, Dr. Yu has combined these really amazing single cell technologies to learn about what an effective immunologic response looks like in terms of the reservoir. And they've discovered this sort of, the residual reservoir is this silent, deep reservoir that is in places in the genome that aren't likely to reactivate. So it's a great story. They've had a couple publications in the past years. There's more stuff coming. So I would recommend that plenary. I'm sure she will provide it in a way that many of us, myself included, can digest better than the most technical versions.
Katie, is that the one that basically Lorene Willenberg came out as a component of? In other words, her reservoir, wherever it was embedded, was so deep that it basically isn't expressing, and so she was indeed cured without medicines?
So I think she was one of the patients that one of the participants that they characterized that had the smallest reservoir. Yes. So there are individuals that are the little dots on the graph that are so exciting to the scientists. Then the people say, that was my dot. Anyway.
So Katie, sorry, you just said, you talked about single cell technology, which is like single cell genomics, and maybe people on the call may not know what that means.
Yeah, so what they did was they paired a whole bunch of really fascinating technologies where they can sort cells down to individual cells, and then they can characterize the cell in terms of what's that cell's phenotype, what type of cell is it, but they can also sequence the provirus, the HIV that's integrated within the genome, and they can look at whether that virus is, where is it integrated within the host, and is it an intact virus that could replicate? So they paired all of those things together in order to characterize the reservoirs in about 60 elite controllers and about 40 people living with HIV on antiretrovirals, and compared those. And it's just, I think, one of the most amazing studies in the past couple of years in understanding the reservoir. I hope that was a little bit clearer. It's really complex technology, but I think they're going to be able to give some sort of broad strokes information about what happens if you have an effective immune response and you get down to this tiny reservoir. What does that look like? Very briefly, Marina Kasky is going to be giving another plenary about HIV BNABs, and that's going to be from prevention to treatment to cure. But she's one of the leaders in the Rockefeller in their clinical trials unit with the Michelle Nussenzweig lab. And so I think that's going to be really interesting for all levels of sort of HIV treatment, prevention, cure. There's a seminar about the central nervous system and infection, inflammation, as well as reservoirs in that. So that is one of the on-demand, I don't remember what day it is, but it's one of the on-demand symposium. And there's 4 really excellent speakers who are going to be talking about all different aspects of infection in the brain and what that means for people's neurocognitive function, what that means for cure, what that means for all sorts of things. I think that's a really great seminar. And then finally, there is a seminar on Tuesday that's on demand, looking at why are CD8 T cells, why are cells that are supposed to identify the reservoir and clear it don't do it very well. So there's 4 speakers, Liddy Troutman and Afam Okoye. They're both from Oregon, and they have really interesting information on people who started antiretrovirals early, and then looking at the key tissue reservoirs of the virus. And then Brad Jones from New York is going to talk about why these CD8 T cells don't seem to be able to clear virus very well. And then finally, Beatrice Hahn, my colleague at Penn, is going to be talking about a study that we did together looking at the virus that rebounds from treatment interruption. And it shows signs that there's this huge innate immune, this interferon response that virus has to get through in order to reactivate. And it may be a reason why the viruses that rebound look a lot different than the viruses that we characterize in the reservoir. So those are the 4 sort of sessions beyond all of the oral abstracts and symposia, or sorry, science spotlights that I think are amazing. But those are the 4 sort of sessions I would recommend for cure.
Awesome, thanks.
I just follow up on the first one you mentioned. There's also, people can look out for a study from Matthias Lichterfeld's group. It's just 3 post-treatment controllers. But they found that there's maybe something similar going on to that elite controller story that Dr. Yu has, where these 3 long-term post-treatment controllers, the reservoir cells that are left, the virus seems to be stuck in these regions of the genome that it can't get out of.
So that's like the same group. Matthias and Shu are lab mates and husband and wife.
Okay.
But yeah, so it's just, they have a whole bunch of studies that have shown this. Yes.
Okay, cool. So it may be something in addition to the elite control, that it's something similar can happen in post-treatment controllers, where their immune system kind of trims away the cells that are most likely to have rebounding virus, and what they're left with is virus that's stuck in places that it can't get out of. And I think I posted some of the other abstracts that I've run across in the chat that might be of interest on sex differences and related to HIV cure. And I think there's one of the abstract selected for the press conference is in a therapeutic vaccine study where they may have a little bit of efficacy of some improved immune response to the virus. I'm not sure how compelling that's going to be, but it's going to be one that's in one of the press conferences out of Spain from Beatrice Moth.
Awesome. Michael, did you want to take it?
Oh, I just wanted to point out one of the other things that I thought was probably one of the more impactful conversations that I had at our CURE workshop was the discussion at the end of everything on Friday, which we had Adam Castillejo, the London patient. We had Gary Steincall. We also had Matt Sharp, who's a gene therapy cure-related clinical trial participant. And we were talking about coming out as research participants and what that means for them. And it was a really, I think, very moving part of our conversation. I sent the link into the chat if people were interested in just hearing that part of the conversation. I just always think it comes down to the people who get involved in the trials that really makes research what it is.
Thank you. I definitely want to watch that. I really appreciate that.
Yeah, definitely. That's all I wanted to add.
Well, thank you, friend. I think so for me, I'll round out the discussion and then I'll thank in particularly us West Coast folks and other folks who are well beyond reasonable hours of operation for coming on today's Zoom session and engaging with the conference at large. But for me, I'm going to focus on weight gain specifically in the context of the switching ARVs. I'm going to focus on anything sex-based differences. That's like my sweet spot. And then I'm also going to do all things cure in the context of ATIs. And then I may round it out with some conversations on health equity and disparities. I noticed at today's plenary, there's some COVID-19 flavored stuff. And they have they have a speaker from Harry who's going to talk about what that looks like, right? So when you talk about this thing or the disparities that we see in COVID and African-American folks, just, the wide disparities there and what the health equity implications of that all are. So I asked individuals on this conversation, what does it all mean? Why is it important when we make these broad stroke statements about including, racial, ethnic, and sex and gender minority populations in these trials, right? That's where the rubber meets the road in terms of really pivoting and lessening the gaps that we see in terms of the health-related outcomes. And so we won't get there if we don't have these very critical discussions. I'm excited to continue to challenge folks about what the health equity related implications of their research means to populations that are grossly impacted by some of the, COVID, HIV prevention, like what does it all mean? So continue to ask yourself, so what? What does it mean for those individuals that continue to be marginalized by these pandemics locally and abroad? So that's what I'm looking forward to this week. And I think we have about 40 seconds before we have to hit it and quote, run to the next plenary. Jim, over to
everyone, you get on your track shoes and get ready to run. Or click, whatever the case may be. And for those of you not attending CROI, have a nice day. Otherwise, I think there's many ways to spend a day. Thank you all for being here. It was a really great conversation. And thank you to all of our speakers, Katie, Danielle, Richard, and Michael, and everyone who weighed in with great questions and comments. We'll follow up with the host of a bazillion links that were shared. Hopefully I'll gather them all. And you can expect an email later. Tomorrow we'll be talking about BNABs. I dropped that into the chat. That'll also come in the follow-up in case you missed it. But it's the same time, same beautiful time for West Coasters, 5:30 a.m. Sorry about that. And then our last day will be about some of the new PrEP data. So that'll be on Wednesday. But thank you all so much for being here. Continue to engage. We'll have some updates on what's happening with yesterday, Greg and Fatima's talk really launched a lot of interest in having a CROI declaration in support to ending vaccine apartheid. So be paying attention to that. We'll help share information. And hopefully we'll see you all again at the conference or at the Margarita Breakfast Club, which is the place to be every day. With that, we'll see you later. Ciao.
Thanks, Jim.
Thanks all.
Cheers, y'all.